Hello, everyone, and welcome to today's webinar, From Accreditation to Excellence, Understanding CAP, CLIA, and Quality Systems in Clinical Laboratories. My name is Kevin Flug-Labrutz, and I'm going to be your moderator for today's event. Today's educational web seminar is presented by Labrutz and brought to you by Burning Rock DX. We encourage you to participate today by submitting any questions you may have asked during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You can also use the Ask a Question box to let us know if you're having any difficulties seeing or hearing the presentation. And now, I'd like to welcome both of our speakers for today's webinar. Dr. Daniel Chen, Clinical Laboratory Director and Molecular Pathologist, College of the American Pathologist Lab Inspection Team Leader, and Christina Brown, Quality Manager, Burning Rock DX, College of American Pathologist Lab Inspection Team Member. Welcome to both of you guys. Thank you very much for your time. How are you guys doing today?
Great.
Excellent.
Yes,
well, thank
we're
you
doing
very
well.
much.
We're excited to be here. Thank you.
Excellent. Thank you very much for your time. I'm going to go ahead and turn it over to Daniel, who's going to start the presentation. So Daniel, whenever you're all ready, you can begin.
Hello. Hi, everybody. Good morning, good afternoon, wherever you are. Today's topic be presenting is about accreditation, understanding the CAP, CLIA, and quality system in clinical laboratory. So just a brief outline. Some of this are clinical laboratory 101. Outline is what is a CLIA lab, and what is a College of American Pathology accreditation, and also including the quality management system, precision medicine, and validation requirement. Since this is just some basic background. So the CLIA laboratory stands for the Clinical Laboratory Improvement Amendment, and it's an amendment enacted in the 1988 to ensure accurate and reliable results across the total testing process in the United States. And it is a federal legislation with a jurisdiction generally mostly by CMS, FDA, and CDC. It requires all the U.S. facility testing human specimen for diagnosis, prevention, or treatment of disease. So basically, if the clinical services, including any of this, the CLIA license is required. There's additionally the thing may, including the, so this is for the U.S. Domestic, but however, there's also an international CLIA lab license, which I will discuss a little bit later. The certificate is test complexity based. The test complexity, basically there's three major categories: wave, moderate complexity, and high complexity. The wave complexity is wave license is a simple test with a minimum risk or error. For example, pregnancy test, test done in the physician's office, or basic urine test. For moderate complexity, the difference is the personnel does not require specialized training and experience. And in addition, the test has no interpretive component to it. And the test required the FDA-approved test designate as a moderate complexity. For high complexity tests, that's a realm for most of the new methodology laboratory development tests are defaulted here. It's for the most of the independent reference lab, and in addition, the PCR and NGS testing for in the high complexity category. For certificate, there's different certificate type. Initially, typically, once the application is made to apply for a CLIA license, and while awaiting for the state or the CMS inspection. After passing the initial paper review, the lab would receive a COR, which is stand for the Certificate of Registration. Additionally, and then after that, the lab can choose to go to either two route, certificate of accreditation or certificate of compliance. And certificate of compliance is inspected typically by state. For example, in California, it's under the California Department of Public Health. Certificate of accreditation is it's granted by so-called deemed body, including CAP, COLA, and other accreditation body, and it has a stricter standard. And typically, the inspection is a peer review system every 2 years. I'm going to dive a little detail about the College of American Pathology for the Certificate of Accreditation route. A basic background: the CAP is the world's largest organization of pathologists and provider of the laboratory accreditation. And there are over 23,000 laboratory participate either by laboratory participant, and the peer review inspections every 2 years with a self-inspection alternative year. And in the U.S., there are about 8,000 accredited lab in the United States and also internationally across 76 countries. And so that's the number of lab this accreditation is overseeing. So it covers 21 laboratory discipline checklists, including blood bank, clinical chemistry, anatomic pathology, molecular pathology, etc. They have a customized checklist that's based on the declared test activity. So, for example, if one were to set up a NGS lab, depending on whether there's a wet lab or informatic component, one would need to declare those activity, and accordingly, the checklist will have the requirement for those activity corresponding with the accreditation. And there's also the proficiency test component, and it's basically it's an external quality assurance. It's to be tested for the test once every 2 years, and the inspection cycle, as I mentioned earlier, is once every 2 years, and also require a quality management system. And this is basically the quality management system breakdown by the high-level quality manual and policy, procedure, work instruction, and record in this tier format. Because of a lot of interest in the molecular pathology and NGS testing, so I want to dive a little bit in about the molecular pathology. So for CAP molecular pathology checklist, they are breakdown into subdiscipline, including the HLA, NGS, infectious disease, and NGS, inherited genetic, for example, cancer predisposing genes, such as BRCA, Lynch type of test. For molecular oncology, it breaks down into two sub-disciplines, including the hematologic disease and solid tumor. I want to segue a little bit about the international CLIA. So there is also a CLIA license for lab outside of the U.S. And from the U.S. government's perspective, it is a requirement for the international facility processing human sample collected in the United States. And this is relevant to lab outside of US to participate in clinical trial where the sample is potentially can be originated from US, collected in the United States. Correspondingly, there's also a international CAP, which accreditation, CAP accreditation can be received for a lab outside of the US. I did a little bit of data mining, and so currently there are 800 lab outside of the US that has this CAP accreditation status, and then this is spanning 76 country slash regions. By country, the Saudi Arabia seems to have the highest number of lab, was 156. China has 120 lab that was CAP accreditation. UAE 86, India 53, and Canada was 44. So what are some of the relevancy for CLIA CAP certificate for a lab outside of the US? So this is one thing is that this is a recognition of the quality and standardization of the lab to receive the accreditation status, and additionally there's a regulatory and strategic alignment with other lab internationally. For example, for pharma, certain pharma or contract research clinical trial, this would allow the standardization of the accreditation and quality standard. Lastly, it's a compliant requirement with the testing the US sample that's coming from United States. A little bit further explanation, the clinical trial service oncology for the pharma trial, oftentimes this includes molecular inclusion exclusion criteria. So those are certain clinical trial service that's of a increasing in the market need right now. So oftentimes from the pharmaceutical company perspective, the service selection often requires international recognized accreditation, such as CAP. And so in summary, CLIA and CAP provide the regulatory and professional framework, which is the excellent, required, and active quality management system. So investing in validation, documentation, and staff competency reduce the risk and support the innovation in clinical trial area. And a strong quality system would enable clinical confidence, regulatory readiness, and external partnership. So thanks for this is my portion of the talk. So now I'm going to give the mic to Chris.
Thank you, Daniel. Hello, everyone. As has been mentioned, my name is Christina Brown. I'm the quality manager for Burning Rock Laboratories in the United States. Today, my focus will be on quality management systems, specifically as they relate to CAP and CLIA. So we'll start with an overview of Burning Rock Lab's operational workflow to show you how we implement our quality management systems in our lab to create a stellar experience for our clients. So Burning Rock has two labs. The first one is in Guangzhou, China, and the second one is in the United States in Irvine, California. So our lab was opened in 2014 in Guangzhou. In 2017, we received our CLIA accreditation and CAP in 2019. In 2021, the US lab was able to achieve both CLIA and CAP accreditation. We also fulfilled the ISO 15189 quality standards in 2022. Since that time, we've also developed an intelligent LIMS system, which is fully integrated into our workflow and fulfills the ISO 27001 certification. Since that time, we've been able to implement a quality management system that consists of over 500 SOPs. We go through regular official inspections, both for governmental inspections as well as CAP and CLIA audits on a regular basis. And our pharma clients also have the opportunity to inspect our lab. We've continued to improve our QMS and be as sustainable as possible, including implementing GCPs, GMPs, and ISO 9001, ISO 13485, and ISO 14064 certifications. Our lab is predominantly a next generation sequencing lab. We do several different types of tests. The first is for comprehensive genomic profiling, both of solid tumors and hematological malignancies. So we have two panels where we can look at both circulating tumor DNA and tissue. We have a 168 and a 520 gene panel. Additionally, we do WES, exome sequencing, and whole genome sequencing. We also have the OncoCompass Heme pan-heme panel, which is a 569 gene panel that's used to profile blood deficiencies. We also have MRD testing, minimum residual disease for solid tumors. These are customized profiles specific for our patients. And we have the Oversee panel, which is an early cancer detection blood panel that can detect a wide range of cancers, including pancreatic, lung, liver, ovarian, esophageal, and colorectal cancer. This is an overview of the workflow, showing that we work closely with our clients at each step of the way to give them the best customized workflow that we can. So every one of our pharmaceutical clients has specific requirements for their specific trials, and so we are able to customize our workflow to fit their needs, including starting with a customized questionnaire, then targeting our systems specifically, our assays to meet their needs. We help them with document preparation. We make sure that our file transfer system for sending patient results is secure and follows all federal and CAP regulations. And we stay with them all the way through the workflow, in even doing the end, a self-assessment to make sure that they have everything that they need to submit their pharmaceutical projects. All right, so now let's talk about how we maintain a quality management system. So what is a quality management system? So quality management system, it outlines all of the policies and procedures to make sure that you are meeting your quality policies and your goals. Doing this assures that we are adhering to both the customer requirements and any regulatory standards, be they CAP, CLIA, federal regulations, or the pharmaceutical client requirements. A good quality management system confirms that the checklist is addressed completely. Quality management systems require approval from the lab director. All of the documents in there, especially technical SOPs and validations, are approved by the lab director. And we need to review these documents every 2 years. That is a CAP requirement. Okay, what are the essentials of a good QMS system? So one of the most important things is that it standardizes our quality processes. So we know that every sample that comes in is treated the same way, goes through the same rigorous testing, meets the same quality standards at each level of testing, and it ensures that we're meeting both regulatory requirements and client needs. It should be a dynamic system. So we are looking to continuously improve the process, both as we become better at doing our testing and as the requirements change. So, you should be constantly looking at your quality systems and saying, how can we make these better? And doing this ensures that you are efficiently meeting your clients' needs and that you are also meeting all of the government requirements. It helps you to reduce errors. and get your patient their results as quickly as possible. Okay, so let's talk about how a quality management system should be designed. So this is, again, a CAP requirement. We start with the quality manual up at the top. Your quality manual, that's your what? So that is a broad overview of the goals of your specific lab, how you're going to meet them, what you need to do to make your process as good as possible. Below that are your policies. So policies are still fairly high level documents that define things like your HIPAA policies, your security policies, personnel and training, like record keeping, all of these types of things. As we move down the pyramid, we get into the who, when, where, the procedures. So these are your SOPs, standard operating procedures. So these are where we're more carefully defining each of the requirements along the way. Below that are work instructions. This is your how. So this is a very detailed document that will detail each step that needs to be done during the process. So your work instructions need to be very detailed so that every day when someone comes into your lab, they will perform the test in the same way to ensure that we have compliance and to ensure that we have quality. So if there are any issues, they're much easier to detect. And finally, at the bottom is our evidence. So these are all of our workbooks. This is where we record everything. So as we say in the lab, if you didn't record it, it didn't happen. So each time a test is performed, you're recording the operator, the date, the lot numbers, which instrument is being used, any issues that were encountered during the run, what samples are being run. So yes, the documentation is the base of our pyramid. It is the most important part because it captures what is actually being done in the laboratory. In our Burning Rock lab, we have over 500 SOPs that are used for our different clinical projects. As I mentioned earlier, because our clients have different needs, we customize our SOPs to meet their needs. So it's very important that anytime you're going to change any step of the process, that you have a new SOP and work instructions that guide that process, and that is approved by the laboratory director. Okay, so this just details some of the quality control steps along the way in our process. So at each step of the next generation sequencing process, from sample receipt through to reporting, there are quality checks. So in this case, before the sample comes to the lab, we want to take a look at it. Was it received in time? Do we have all of the information we need on the patient? Is the sample of good quality? As we're actually in our wet lab portion, are we getting enough DNA? Is the DNA good quality? Do we have enough for sequencing? Does it meet those needs? The cluster density, the quality, the Q30 scores, and finally, when we're actually doing our reporting, did it sequence properly? Do we have good results to give to our patients? And then finally, that the report contains accurate information on our patients before we can transfer it to our clients. So again, very important to be looking at each step of the way to see that we're meeting the quality needs. This is another step for the quality control monitoring. So in our labs, we have different teams that will look at different pieces of the quality puzzle. It's important that they are all in communication both with each other and with the clients. So our QA team is in charge of doing the study initiations, maintaining the study records, keeping track of any deviations that occur. Operations teams, they're doing their daily, weekly, monthly checks. They're making sure equipment is functioning properly, that the TRFs, the test requisition forms, are complete, maintaining chain of custody records, and issuing any memos regarding any deviation during the testing. The QC team sort of looks over all of that. They do the assay validation. Assay validation actually is one of the things that I think is missed the most often by laboratories. At least that's what I've seen as an inspector. So we want to make sure that when we're initially validating our assay, that it is meeting all of CAP's requirements. CAP has very specific requirements for validation, and they vary based on your CAP checklist. So make sure when you are validating your assays that you have enough samples and that there are enough different things being tested to meet the CAP requirements. And again, we're keeping our testing records. We are issuing any logs and improvement records for our samples. All right, next up, I'd like to talk about what I call creating a quality mindset. So quality should always be in the forefront of everyone in your lab. How can we make this process better? Are we doing all of these things correctly? And one of the best ways, I think, personally, to do this is through internal audits. So internal audits where we actually go through and we are testing our system. We also use what we call tracer documents, which are test orders. So we put dummy orders into the system just to make sure that we get the results out that we expect and that things are running properly, that we are reviewing things like calibration data, QC, lot-to-lot verifications, and our daily logs. And it's important to trend this data and to look for outliers. So just recording it is not enough. You need to have someone that's actually actively reviewing this information to ensure that it is correct, that you're not seeing any deviations. We use what are called quality assurance monitors, or KPIs, which I will talk about in a later slide. The next is anytime that there is a non-conforming event, be it an equipment malfunction, a sample that doesn't meet the needs or fails one of the steps of QC, that we're documenting it. So this documentation is especially important to have. If there is any incident that causes patient harm or has the potential to cause patient harm, it needs to be reported immediately. And so the severity of the deficiency will determine the action. So we need to instill in our laboratory staff that all of these things need to be recorded. And then quality and the laboratory director can decide what the next steps will be, whether a CAPA needs to be opened or there needs to be some reporting to the local agencies. And then also that we have a plan of correction. So anytime we have a CAPA or we have an inspection finding or citation, that we have a plan of correction. And quality needs to ensure that these are actually being reported, that when we have deficiencies, that there's a plan of correction to fix that deficiencies, to improve our systems so that things like this don't happen again, and to ensure that that improvement actually, fixes whatever the problem is. Next up, we'll talk about daily operational practices. So these are things that are done every day by the lab to ensure continuity and transparency. So starting with logs and records, that we have our bench cleaning records, that we're doing environmental monitoring, equipment maintenance, and that all of that is being recorded and again, reviewed. Not just that you're writing it down, but that someone is actually looking at it and reviewing it. Continuity. So if you have, say, 2 shifts and a procedure did not get finished, that we have a way of transferring that information to the next team to know exactly what step they are in the process, to know if there have been any problems. So we need to have what's called a hands-off procedure. We also need to ensure that we have a chain of custody records. So anytime a sample is transferred from one part of the lab to another, or from one lab to another, that we have a record of that. That includes what is being transferred, who is in charge of the transfer, where it's going, and that we follow up to make sure that it actually makes it to the next place. And this ensures timely and accurate responses. And finally, that we have transparency. So we want to encourage transparency. So when there are incidents in our lab, we want to make sure that they're not punitive, that we're not punishing people for anything going wrong, to encourage them to actually maintain these records and know that they have the support of upper management if they do need to report something. So we want to empower our team to take ownership. All right. Next up, let's talk about the different phases in the laboratory and a little bit more about key performance indicators. So there are 3 phases in a clinical lab: pre-analytic, analytic, and post-analytic. So your pre-analytic phase, this is anything that happens before the samples make it to the lab. So this is sample collection, labeling, transport. This has to do with the contracts that you sign with your clients. The next step is the analytical phase. So the analytical phase is anything that happens in the laboratory. So this is the sample testing procedures, sample analysis, basically, yes, anything that the lab is doing. And then finally, the post-analytical phase. So this is where we're doing result reporting and sample storage. So ensuring that the samples are stored properly for the correct amount of time, that our records are being stored properly, that we are meeting whatever those requirements are for the length of storage and the conditions of storage. So our key performance indicators are things that we can look at in each phase in the lab to make sure that we are actually doing what we need to be doing. So for example, your pre-analytical KPIs might be things like your rejection rate, how many samples are misidentified, if there are any improper shipping. We need to keep track of how many samples meet each of these key performance indicators. In the lab, we look at things like competency assessment. Is everyone in your lab properly trained? When you're running your proficiency, your PT samples, that you are actually getting acceptable results from those, and that your equipment maintenance is up to date. For post-analytic, these are things like turnaround times, customer satisfaction. Right. So key performance indicators is something that CAP requires us to be looking at. I think it's important to set goals for your KPIs. So each time that you set a KPI that you're going to review, you need to say, okay, we want to hit, for example, turnaround time at least 95% of the time. These need to be reviewed by the lab director at least monthly. So we usually do it as part of our monthly quality meeting. We look at the KPIs for that month. And we need to make sure that we are trending these and investigating outliers. So if you, for example, start to see that all of a sudden your turnaround times are not being met, like month after month, that's an opportunity to look into that and to see what's going on. And anytime you have an outlier, there needs to be an investigation as to why that was not met and what exactly happened in that case. Okay, and next up, we'll talk about maintaining quality records. So these are all of the different types of quality records that we need to keep. There are specific requirements for how long certain records need to be kept, how long you keep your workbooks, how long you keep personnel records, how long you keep your PT records. So these are all listed in the CAP checklists. So I suggest just copying them directly from the CAP checklists into your SOPs and making sure that you're actually meeting those. So we have the SOPs that we've already mentioned. We also have instrument repair and maintenance. So anytime maintenance is performed on an instrument, we need to make sure we have a record of that. All of our calibrators should have records. Environmental monitoring. This is one where I actually see a lot of labs have problems with environmental monitoring. So make sure that you are in the temperature range that is required by both your reagents and your SOPs. Make sure your environmental ranges are defined. Proficiency testing, you can order PT samples, proficiency testing samples directly from CAP. If your CAP activity menu is up to date, they will tell you exactly what proficiency testing samples you need to order, and just make sure that you are actually running those and that someone is reviewing them. Personnel records, again, CAP has very specific requirements for what you need for each of your personnel. Make sure that they are in a personnel file, and this should include both competency and training records, as well as things like their CV, their licenses if they are licensed, etc. And quality assurance monitoring, we've already talked about KPIs and trending. So one of the key components of an effective QMS is to maintain these quality records. So I've already discussed internal audits a bit. Internal audits are required by both CAP and CLIA and most ISO requirements. They should be done at least annually. Annually seems to be, that I think that's a good amount of time to do it. And they should assess all of the key components in your system. So you should be looking at all of the different parts of your system and make sure that things are going well. And this helps us to increase our accuracy and efficiency and also to detect any fraud or errors. Okay, so finally, some key takeaways from today's presentation. Inspection readiness depends on a collaborative team consistency. So again, make sure that the different parts of your laboratory are talking to each other and that they feel empowered to report things like errors or inconsistencies. Recognize that laboratory incidents will happen. Like even in the best of labs, there are going to be things that go wrong. There's going to be equipment malfunctions. There's going to be mistakes. Recognize that those will happen. Correct them. So come up with a plan of action and use that to improve your system. And do your best to encourage a quality mindset for your people. Okay, and so with that, I'd like to thank you for your time, and we'll go forward with the questions. Thank you.
Well, thank you both very much for your informative presentation. We're now going to begin the Q&A portion of the webcast. If there are any questions that you would like to ask, please do so now. Please just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. And I'm going to bring back on Dr. Daniel Chen. Thank you very much for your presentation. Really thoroughly enjoyed it. And we got a lot of great questions already starting to come in, so I'm excited to dive into these. The first question that came in, based on your experience, what are some of the most common compliance gaps you see, especially when labs outside the U.S. try to align with CAP or CLIA requirements?
During my inspection experience, or what I often find is the number one gap is the reported activity manual which reported to CAP, and then what's actually practiced. For example, a lab that's doing NGS and for oncology and also doing hereditary cancer, but they only reported the oncology test menu versus the hereditary cancer, so that's something occasionally you can find, but that's one of the bigger gap, but it's actually pretty simple from the inspector perspective, it's simple to verify that, because of you can go to the website or to check the test ordering menu to verify that.
Okay. Excellent. Thank you very much. Next question, I think, is a little bit of a follow-up with that, speaking from U.S. and international considerations. For international labs, why is it important to understand CAP and CLI standards, especially when working with molecular or NGS testing?
So oftentimes the international lab requests the CAP accreditation is for business need, because CAP accreditation is a widely recognized international standard. For example, if the lab's business are to do oversee pharmaceutical contract research business, the pharma CRO pharma client oftentimes are familiar with such standard, and it makes their inspection audit, the pharma audit of the lab much straightforward, because their internal QC people understand this requirement. So it's a great way to get business, because they would your potential client would understand the standard.
Okay, excellent. Once again, a lot of great questions starting to come in. Once again, if you do have a question you'd like to ask, just type your question into the Ask a Question box and click Submit. Next questions coming in will be Daniel, if you could give one piece of advice to laboratories around the world striving for higher quality and compliance, what would it be?
So the quality excellence is actually it should be a corporate culture that should be from the top to the bottom. So the I would advise the management, the top management, to support that, and then that value should be instilled from the top to the bottom. Because the reality is that oftentimes it's a little bit of a tug of war between achieving high quality, that's for the best patient care, versus the finite resources. So the quality team and the system would do well if this need is recognized by the upper management, and then the communications bring down to the bottom.
Okay, excellent. Sifting through, let's see if we have any other questions that came in. Got one that says, if a lab performs a test in multiple departments, so hematology and blood bank, are both required to participate in proficiency testing or alternate assessments, or is it only required once per CLIA number?
Okay, just depending if it's the labs in the U.S. or in the international. I'm just using the U.S. as an example. In United States, in order to do the clinical testing, you need to have the CLIA number, CLIA license, and then every activity, every clinical test under that CLIA license need to be reported to CAP for the compliance need. So it's pretty straightforward. So whatever is under that CLIA number that you're doing the clinical testing, those are fair game as part of the CAP accreditation.
Okay, excellent. Looks like we have time for one more question. Question asks, if samples are sent from one lab to a reference lab, which lab is responsible for assessing the specimen acceptability or rejection criteria? For example, if a blood sample was received by one lab and referenced out to another, which lab would assess the sample for clotting before testing?
It would be both. Let's simplify this scenario. Lab A accession the specimen and send it to Lab B, and then the B does some wet lab testing, and then the result go back to Lab A for sign out. So the validation should be done by Lab A, and to ensure the every piece. But if the sample is sent from A to B, the B also need to assure the adequacy or of the specimen to be accepted.
Okay.
Well, I'll say both. Yeah, short answer is both. Yeah.
Sure. One more did sneak in here. If an international laboratory holds an ISO accreditation, is it still required to obtain a CLI certification to participate in clinical trials involving the U.S. or to test U.S. patient specimens?
Yes, because the international CLIA is the purpose is, for international lab, which means outside of U.S. to test the sample that originated from U.S. So theoretically, you can have an international CLIA without the ISO, but if it's an international lab that are testing U.S. sample, they definitely need the international CLIA.
Sure. Excellent. Lastly, are we required to retain paper copies of control and maintenance logs after they've been scanned into the computer?
You're required to retain any copy. You know, I would my assumption is the electronic copy is sufficient, but it doesn't hurt to keep the paper trail just in case there is some audit going on.
Sure. Excellent. Well, thank you again for your time today and your important research. I also want to thank LabRoots for hosting this event and for Burning Rock DX for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting presentation or for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So once again, thank you very much for your time. And until next time, goodbye.